Hi everyone, my name is Guangyang Hou. I'm a senior engineer from APC Limited. Welcome to my webinar. Today, my topic is novel strategies to transform the pace and cost of high-potency API development. This is an overview of this webinar. First, I will give you some background info on high-potency API, including its definition, and high-potency switch capabilities at APC, and high containment operation. Second, I will introduce chemical engineering strategies applied at APC to handle high-potency APIs, which will include continuous processing approaches and model-supported high-potency API crystallization process design. APC is a contracted research organization based in Dublin, Ireland. We have over 200 chemists and engineers working across a broad range of areas, including small and large molecules. APC collaborates with pharmaceutical and biopharmaceutical companies across the globe to accelerate the delivery of medicine to the patient. What is high-potency API? In general, an API is classified as highly potent if it has an 8-hour average occupational exposure limit of not more than 10 micrograms per meter cubed. Based on the volume of occupational exposure limit, API can be categorized into 5 bands, with band 1 being minimum or low toxic, while band 5 being high toxic. For different bands of APIs, different containment strategies and engineering controls are needed, especially for band 5 material. Due to its high toxicity, high containment operation is needed. APC has 2 high containment labs. In each lab, there are 8 to 10 film hoods, 6 to 8 isolators, and glove boxes. We have the capability to run multiple high containment projects in parallel. Our high containment lab has 4 analytical capabilities, including HPLC, Marvin, DSC, XRD, and microscope. The high potency API prepared in the lab doesn't have to be transferred to an analytic lab. This would avoid the potential cross-contamination. Here, I'd like to introduce 2 rigs that have been built in our high containment lab. The first rig is the rig for wet milling, as you can see from the photo on the bottom left, where EasyMax Workstation 402 together with ICA Magic Lab were built in the same glove box for part size control. This is particularly useful for delivering small part size with narrow part size distribution. However, leakage is always a concern for wet milling. The use of a glove box to contain both equipment would provide a safe working environment for handling high potency API. The second rig is a rig for antibody drug conjugate reaction. Due to the high toxicity of payload, conjugation reaction has to be carried out in high containment lab. Payload and all other species were prepared in glove box and contained in Durand bottles, which were then placed on top of balance. The mass flow rate of each species were recorded and monitored throughout the reaction. To enhance the mixing for reaction, 3D printed static mixer were applied And a flow cell was assembled at the outlet of the flow reactor to accommodate FTIR or inline UV in order to track the concentration of interested species. In high contaminant labs, over 50% of lab work has to be conducted in glove box. The high safety standard and challenging operation using glove box definitely slow down the pace of process development. On the right table, we summarize the time and resource needed to complete a series of activities for high content projects against typical projects. In general, for the same task, sub-resources with even longer time are needed to complete a high content project against typical projects. It's apparent that high safety standards requirements and extremely challenging handling procedures require more time and resource. Labor-intensive trial and error experimentation approach would always mean high cost and extended timeline. At APC, we always apply model-supported approach to assist process development. This is even upgraded to a high level for high content projects in order to minimize the human to compound contact time. We had one distillation project where constant distillation endpoint was required to facilitate the subsequent crystallization process. The traditional approach is to sample throughout the distillation process until the desired thermal composition was reached, while Stylochem thermal swap model could be employed to accurately predict the thermal composition across the distillation process, through which we were able to know the distillation endpoint. This approach significantly reduced the experimentation time. In addition to modeling, we also apply continuous processing to our high content projects. For continuous process, it features high productivity to deliver the same quantity of material. A much smaller reactor is needed to handle smaller equipment would be much safer. In addition, continuous processing doesn't need or need much less human intervention, which is much safer in handling high potency API. We have two continuous platforms, including MSMPR and PFR. MSMPR is suitable for high-potency APIs with relatively slow kinetics. This platform is typically applied to deliver metastable form, to purge kinetic impurity, and to attempt modified crystal. PFR is suitable for high-potency APIs with fast kinetics. This platform is mainly used for delivering small particle size with narrow particle size distribution. And this is an example of our MSMPR platform, where a two-stage MSMPR continuous cooling crystallization rig was built to control the crystal. As you can see from Dry-to-net photos. The first swimsuit has two feed tanks, two feed lines, and two feed pumps. The feed solution was charged to both tanks at the start of the experiment, and no human intervention was needed in this swimsuit throughout the process except for turn on and off the feed tanks. The second swimsuit has two-stage MSMPR. The first stage MSMPR was combined with wet meal for particle size and heat control. As you can see, there are two wet meals. The second wet meal was used as backup just in case if the first wet meal stopped working. We didn't have to mess up the process. The slurry transfer was automated using a programmed control box. Filtration system in the third swimsuit was also automated through the control box. Once the steady-state operation was reached, the process was monitored using PATs until the desired quantity of material had been generated. No human intervention was needed at all. And this slide exhibits an example of PFR. As you can see, jet milled quality particle size was delivered using our PFR rig. Both the particles were generated in wet conditions, which is much safer when compared to size reduction in solid state using jet mill. Finally, I'd like to give you a case study on model-supported high-potency API crystallization process design. And the current particle size isolated through the baseline process achieved a D50 of over 100 micrometers, which is much larger than the desired particle size, with D50 of less than 10 micrometers. And the model compound is unstable during milling. Therefore, the objective was to design a crystallization process to deliver the target PSD directly without size reduction. At APC, we have our own workflow for solvent selection. We typically start from 70 solvents by using Cosmo RS and Material Studio, and based on the solubility result, predict solubility result, and solvent propensity and nucleation behavior, and 6 to 8 solvents can be identified as potential good and anti-solvents, which were then brought into lab for 2-point solubility measurement. So this 2-point solubility measurement will inform the yield, productivity, and also the wet cake or dry cake can be analyzed for purity and foam check. And finally, the most promising 2 to 3 solvents will be examined at a crystalline scale, through which further info such as metastatic zone width, crisp form, purity, crisp heat, process yield, and productivity can be examined. Up to this project, we screened about 35 solvents. As you can see, the Cosmo predict solubility ranking at 25 degrees. And this API suggested a relatively high solubility in all the high boilers, while it exhibits much smaller solubility in water, heptane, and some other solvents. Based on the solubility ranking, were chosen, we choose several good solvents, such as acetone and methanol. At the same time, several alternative anti-solvents were identified, including water and heptane. In order to choose the most appropriate solvent to enhance nucleation, activity coefficient of the API in different lead solvents and biliary solvent mixtures were estimated using Cosmo. An activity coefficient suggests the interaction between solvent solute against solute solute. In general, a small activity coefficient such as in glucosome 6 and glucosome 1 would suggest much stronger solvent solute interaction against solute solute interaction. In such some system, the nucleation is quite difficult. While on the other hand, with a larger activity coefficient such as in water and antisolvent 1, it would suggest a much stronger solute-solute interaction, and it's much easier to get nucleation in such some system. And for biliary solvent mixtures with an increase in water or anti-solvent, the nucleation difficulty was eased. Even though water and antisolvent 1 exhibit the highest nucleation propensity, however, due to their limited solubility, biliary solvent mixtures were employed. In this study, as you can see, based on the order of the nucleation difficulty, the top 3 biliary solvent systems, including water glucosome 1, water glucosome 2, and water glucosome 3, they were employed for the subsequent nucleation trials. The solubility of the API in the above-mentioned 3 biliary solvent systems was measured, and the mass-equal post-solubility measurement was charged to a predetermined water and hold overnight. This was to examine if nucleation would occur and what the associated HPAPI and size-like. As you can see, the microscope images taken in the nucleation trials in glucosome 1 and water solvent system, significant agglomeration was present, and therefore this solvent system was discarded. And in glucosome 2 system, even with the presence of agglomeration, it seems like different crystal morphology was isolated. Therefore, this solvent system was kept for further study. And the most promising solvent system was glucosome 3 water solvent system, where discrete lead-like particles were isolated. Before we started process development at EasyMax scale, solubility phase diagram was fully mapped out to determine the appropriate crystallization starting and end point. For solubility measurement, we typically measure solubility in 3 different temperatures, and at each temperature, we measure 3 to 4 solvent compositions. By using DialChem solubility regression utility, we can fully map out the solubility in the interested solvent composition ranges and at different temperatures. So based on the shape of the solubility curve, if synergistic effect is present, the corresponding maximum solubility can be used as crystallization starting point. And based on the sensitivity of the solubility to the anti-solvent composition and temperature, it's possible to identify the potential crystallization endpoint. After the crystallization starting point and end point had been determined, we designed the crystallization process based on the solubility curve. First, prepared saturated API solution in predetermined starting foam composition at room temperature in reactor 1. And second, pre-filled reactor 2 with anti-solvent. Third, charged API solution to the crystallizer containing anti-solvent to reach the predetermined crystallization endpoint. Samples were taken throughout the API addition period to examine the crystal habit and size, and the same procedure was applied to both bioluminescent mixtures, and only one of them suggests most promising result and therefore pursued for further study. And to enhance nucleation while suppress crystal growth, the following three factors were evaluated, including API solution addition temperature, addition time, and activation speed. And a similar sampling approach was applied to examine the crystal habit and size. And after the three more rounds, we managed to deliver the desired particle size distribution. So as you can see, throughout this project, only 5 process rounds were carried out. A summary on the case study. APC utilized a novel in silica workflow which guided suitable solvent selection to enhance nucleation and restrict crystal growth. Solubility phase diagram aided crystallization process development, enabled near right first time design of a revised crystallization to deliver API of jet particle size specification. And modeling supported high-potency API crystallization design substantially reduced the time frame and workload enough as per the typical extensive trial and error experimentation approach.